Got it.
Listen, I didn't really get to finish yesterday's lecture, so I'm going to start out with the end of chapter 5, 5.3.
Are we doing 5.4?
We're not doing 5.4. You can just skip 5.4. They took a lot of stuff off the AP exam because a lot of teachers were complaining they couldn't finish the book. So
computers can do that?
we won't worry about 5.4. I just wanted to show you a few of these videos of bulk transport, which are good videos that you didn't get to see.
Should I watch the video?
Huh? Yeah, you can point it. Whatever you think's important, you follow. So is this what still confuses y'all, the cells?
I just thought when you would place something in a hypotonic, I guess I say it, solution, like the solution would take a lot of water.
No, hypotonic means it has a lot of water in the solution.
Okay.
And so it's going to move elsewhere. Yes?
So wait, does tonic, when you're saying hyper, does that refer to the mixture or the water?
It refers to hypertonic. Yeah, it refers to the water. No, yeah, it refers to the mixture.
Okay, so hyper means a lot and hypo means not a lot?
Hyper means a lot. And tonic, I guess, is referring to the solute. A lot of solute. Hypertonic solution has a lot of solute.
Why is the animal cell burst with the plant cell?
Because the plant cell has a tough cellulose cell wall around it that won't allow it to burst.
Is that why when you overwater something?
What's that?
No matter how big you get it.
Yeah, it'll never burst.
Does it stretch?
It stretches, but it doesn't burst. What were you saying?
Is that like when you overwater something?
Yeah, if you put a lot of water in a plant, the plant will stand really stiff because every cell is kind of expanding.
So hypotonic has more water on the outside of the cell and more solution in the inside?
If a cell is put in a hypotonic solution, the word refers to the solution itself. Don't worry about the cell, just worry about the solution. If the solution is hypotonic, it's got a lot of water in it. If the solution is hypertonic, it doesn't have much water in it. So let's take a cell and put it in a hypotonic solution. That's like pure water. What's going to happen? Water's going to rush into the cell.
So
it moves towards the salt.
Yeah, the water always moves toward areas where there's less water. So this is showing a cell placed in a hypotonic solution. Water will rush in.
And solvent is where you put salt.
Yes.
Solvent would be water, and then the solute.
Solvent is water, solute is sugar or salt or whatever dissolves in it. Other questions about movement of water in cells? Again, that's our lab, Friday, Monday, and Tuesday. We're in the lab working on this. So it's good to have a good understanding going into the lab. Let's see some bulk transport.
So is that like all osmosis?
Osmosis is movement of water. That's correct. Let me ask you this. Why not water your plants with salt water? There's a good reason.
Because
Water,
salt in the water, there's more salt in that water, so the water will actually leave the plant and go into the salt.
That's exactly what would happen. If you water your plants with salt water, try this the next time your folks go out of town. Bring some salt water and water all their plants with salt water. The salt water will get into the soil, and the plant itself has more water in it than the salt water does. So water will rush out of the plant and into the roots where you put the water. And it'll shrivel up and die. This is the same reason why you can't drink salt water if you're lost at sea. The water will rush out of your cells and into your stomach and intestines where the salt water is. instead of reverse. And you'll dry up and die. They'll find you on your little raft all shriveled up. Terrible way to go. Oh, we saw this one already. Remember this one? Exocytosis. This is probably the best video showing it. A little bubble comes out of the Golgi and the substance is released. Here we take in some large particles. What's this called?
Endocytosis.
What type of endocytosis?
Phagocytosis.
This is phagocytosis. Phagocytosis. Huh? You like that? You like phagocytosis? We saw that one. That wasn't that great. Oh, here's a paramecium. What's he up to?
Protists, such as paramecium, sweep food particles into their gullet, trapping the particles in a food vacuole. Digestion takes place within the food vacuole, and the products of digestion pass through the membrane of the food vacuole. Finally, the food vacuole merges with the plasma membrane, expelling waste products into the environment.
That was awful. There's the expelling of waste products. That's like going to the bathroom with paramecium.
In phagocytosis, phagocytes are attracted to the area of invasion by chemical products of the microorganism, phospholipids released by injured mammalian cells, or by components of the complement system. The phagocyte moves into the area of invasion and then attaches to the microorganism. The attachment is mediated by a variety of surface receptors, including antibody, lipopolysaccharide, and complement receptors. For example, the C3b component of complement coats bacteria or other particles and then binds to C3b receptors on the phagocyte. This process of coating to enhance phagocytosis is called opsonization. The microorganism is then engulfed by the phagocyte into a vacuole known as a phagosome. Vesicles in the cytoplasm called lysosomes fuse with the phagosome, releasing digestive enzymes such as lysozyme and proteases into the phagosome. The structure resulting from this fusion is called a phagolysosome. Inside the phagolysosome, microorganisms are killed and digested. Finally, the digested contents of the phagolysosome are eliminated from the phagocyte by exocytosis.
That's how your white blood cells kill things. Wasn't that good? You getting all this, Sarah? You don't sound too interested in phagocytosis.
Phagocytosis, which means cell eating, is a form of endocytosis. A cell such as a phagocyte engulfs surrounding material, forming vesicles called phagosomes, derived from the plasma membrane. Phagosomes merge with vesicles called primary lysosomes that contain digestive enzymes, forming secondary lysosomes. Digestion takes place within the secondary lysosomes.
Okay. I guess we get the idea. Penocytosis is taking in small particles from the environment. Often they're water droplets.
Penocytosis, which means cell drinking, is a form of endocytosis. A cell brings in droplets of the environment by pinching them off with the plasma membrane. The pinocytic vesicles break away from the membrane and move to the cell's interior.
So it can take in a lot of water that way. Notice the plasma membrane will be getting slightly smaller while doing this. The cell has a way of making more membrane if it wants to.
Are we going to learn about that?
We already did with the Golgi apparatus and such.
No.
That's what goes on there. We didn't really talk too much about it.
Cells also use energy to transfer materials in bulk, but this time by forming membranous sacs that hold their contents under wraps. In a process called endocytosis, part of the surface membrane encloses the material, forming a sac which brings the contents into the cell's interior. In the opposite process, called exocytosis, the sac moves through the cytoplasm to the membrane, fuses with it, then releases the contents.
Oh, that was cool. I like the music. Finally, there's this process called receptor-mediated endocytosis, where there are little proteins that receive a signal. Little solute molecules will stick to the proteins. The proteins sit on the surface here. The solute molecules stick to them. And then after the solute molecules bind with the receptor protein, the whole thing pinches inward. So it's called receptor-mediated endocytosis because the receptor proteins are helping the endocytosis. Receptor-mediated.
The substances used as fuel by single-celled organisms include...
Oh, here we go.
If the material is a liquid, it is called pinocytosis. Some types of molecules, such as low-density lipoproteins, LDLs, are transported across the plasma membrane by receptor-mediated endocytosis. These molecules first bind to specific receptors embedded in the plasma membrane. The receptor molecules are concentrated in an indented pit coated by the protein clathrin. When sufficient target molecules accumulate in the coated pit, the pit deepens, seals, and is incorporated into the cell as a coated vesicle. Exocytosis is the reverse of endocytosis. This process results in the discharge of materials from...
What's the protein called?
The protein is called clathrin that coats the pit. I don't know if that's in your book or not.
So what happens to that protein when it gets to wherever it needs to be?
What happens to the clathrin?
Yes.
I don't know.
Exocytosis is the...
I don't know.
Is the membrane like recreated?
I don't know. That's a higher class than I teach. Take that second or third year bio students learn all that stuff. Sure, I learned it in college. Quickly forgot about it when the next football game came around. Okay. We good with this? Y'all okay? All right, let's go on to the next